Professor Vallortigara is, as I said, University of Trento and graduated at the University of Padua and is now 130 kilometers away from there. In a career spanning over 30 years, he worked extensively on brain lateralization and what you could call comparative studies. He's had numerous books and articles published in journals like Nature and Science and Cognition and Current Biology. And without further ado, I would like to ask Laura to start playing his presentation. So everyone, enjoy.
Good afternoon, and thank you very much for this kind invitation. I'm really very honored and very happy to take part in this conference. So thank you very much to the organizer for inviting me. The talk will be roughly divided into two parts. During the first part, I summarize quickly the work we did mainly at the behavioral level concerning the existence of animacy detector mechanisms in young organisms such as newly hatched chicks and human newborns, devoted to reveal the presence in the environment of living creatures. And in the second part, I will discuss more deeply the neural and neurobiological mechanisms underlying this animacy detector as the foundation, the basis of our social brain. So let's try to share the screen. The topic I'm going to discuss is deeply rooted into the tradition of classical ethology, and it is associated with filial imprinting. I guess you are familiar with filial imprinting. This is a historical movie showing Konrad Lorenz with his birds exhibiting the classical following response. Actually, Lorenz was not the scientist who discovered filial imprinting. Was discovered by Douglas Spalding. He mainly popularized the phenomenon. And imprinting is the learning process by which the young of some animal species, particularly of precocial species, learn to recognize their social partner, their mother, by simply being exposed to it for a very short period, soon after hatching or soon after birth. And in the laboratory, imprinting is usually studied by exposing chicks to a conspicuous, to a salient object, such as a rotating red box, and chicks are quite happy to run the equivalent of kilometers in order to rejoin the object they have been exposed to. And quite interestingly, as a result of this exposure, they also develop a recognition memory. And when they are presented, for instance, with a novel, unfamiliar object, such as a red rotating blue cylinder, they refuse to approach to show the following response to the unfamiliar object. And we learned quite a lot about the neural mechanisms of this kind of recognition memory, thanks to the work of Gabriel Orn and Patrick Bateson and other people in Cambridge. And they found that there is a small semi-lunar region here in the forebrain of chicks, which is the AMM. The acronym is for intermediate and medial part of the mesopallium. It is an associative area, sort of cortical area in the avian brain, which is important for imprinting. And if and in this area, for instance, there are neurons that following exposure show selective response, higher levels of spikes, frequency of spikes to the particular object they have been exposed to, but not to a novel object. And of course, if the AMM is lesion or inactivated by pharmacological agents, for instance, chicks appear to be amnesic. They seem to be unable to recognize by approach responses the original imprinting object with respect to an unfamiliar and novel one. But several years ago, in the laboratory of Gabriel Horn, a very strange and weird observation was carried out. Namely, considering together the results of several experiments, they found that if chicks were imprinted on artificial objects like, say, rotating red box, blue cylinder, and so on, indeed, as a result of the lesioning in the AMM, they were amnesic. But if imprinting occurred onto naturalistic stimuli, such as, for instance, this sort of stuffed hen, it is actually not a domestic hen, it is the jungle fowl, so the ancestor of the domesticated species. Well, in this case, still a preference for the imprinting object was apparent. And that was quite disappointing at the start, because it seemed to suggest that there was some mistake, some error, and that AMM was not actually the site of the memory. But then they suddenly realized that imprinting has nothing to do with this preference, because when testing newly hatched, completely naive chicks for their spontaneous preference before any imprinting has occurred, for, say, a rotating red box and a stuffed jungle fowl, a preference for the hen was apparent. It was not a spectacular, exceptional one, it was about 65%, but quite consistent and significant. So in the following years, Mark Johnson, Johan Bolwis, and others tried to understand what is special about the appearance of a stuffed hen that make it so attractive for young chicks. And so they prepare scrambled version in which the shape, the overall silhouette of the stuffed hen was changed, but no preference was observed between these different versions. They were identically attractive for the chicks. And also, when pieces, cluster of portion of the stuffed hen were simply attached to a rotating box, still no preferences were observed. Only when small pieces of the original object were attached to the rotating box, such a way that the cluster of certain critical region, and in particular of the region of the head and neck region, were completely disassembled. At this point, a preference for the canonical version with respect to the scrambled one was observed. And this relevance, this importance of the head and neck region was also not limited to the species, because the same preference, or better, the same absence of any preference was there for a completely different species, like duck, for instance, and even more surprisingly, for a species like a ferret, for instance, which is actually a predator for young chicks. Mark Johnson and John Morton tried to conceptualize this data, and they look also a few years later that it was discovered that human newborn seems to have somewhat similar preference for face-like stimuli. human newborn seems to be predisposed to attend to face-like stimuli. And as I said, Morton and Johnson tried to conceptualize this observation, arguing for two mechanisms, a device for face recognition that would comprise a conspec mechanism, that is a predisposition for dirty and fast processing mediated by subcortical brain areas, probably. That was speculation, obviously, superior colliculus, pulvinar, or amygdala. And then a conlearn mechanism that would be associated with learning and would require specific visual processing associated with the role of cortical regions, allowing discrimination of individual faces. More or less in the same years, I was involved with somewhat similar but with a different origin issue. Namely, if you look at the classical ethology textbooks, it is apparent that they say that any kind of stimulus is effective or could be effective for imprinting, but better if the stimulus is in motion, is set into motion. So I wonder whether any kind of motion would be identically effective, or if, for instance, chicks have a preference for the very peculiar movement of a hen, of a chicken. And the problem obviously was to disentangle the purely kinematic aspects, the motion in itself, from the silhouette, the shape of the stimulus. And in order to study this, we make use of a phenomenon very well known in perceptual psychology, namely the use of point light display, which was introduced by psychologist Gunnar Johansson. When you look at a stationary image, you cannot recognize the human figure, apart from the case in which you have a particular talent for projective geometry. But when the stimulus is set into motion, you can immediately recognize a human figure. And also not just the shape, the structure from motion, but also the type of action the creature is involved with, say doing karate, for instance, or playing tennis, soccer, and so on. So briefly, what we did was to present chicks with point light display. These were completely naive chicks, just out from a dark incubator, and they have in the dark some motoric activity in a running wheel. That's important because it's sort of permissive mechanism in order to exhibit this sort of inborn preferences. And they were located into a runway, and the opposite side there was two high frequency screen depicting the same number and the same average velocity dots, and these dots move in a different way, for instance as a walking hand or as a rigid hand rotating around the vertical axis, or with a completely random Brownian motion, or with a scrambled version of the walking hand. So single vector of movements were exactly the same, moving in the same way, but they were completely displaced from the spatial point of view. And therefore, the characteristic of the motion, semi-rigid motion, is there, is still biological motion, but of a completely unknown creature. What we found was, for me, in part unexpected, because yes, we found that there was a preference, this completely naive chicks for the hair walking with respect to rigid and random motion, but no preferences at all between the hand walking and the scramble hand. So the preference was not relative to the movement of the species, and in fact, the scramble hand was preferred to rigid motion and to random motion. In retrospect, this is in some way not so surprisingly, as we will see. Consider, for instance, the the case of this sort of stimulus, this is a cat, which is a predator of young chicks. But when we tested them with cats, we found that the walking cat was preferred to rigid motion and to random motion, but not to the walking hand. So chicks are perfectly happy to approach a point-light display of a walking cat or of a walking hand with the same probability. So again, this may appear surprisingly because of the fact that these are predators, but the point is that the function of this inborn predisposition is obviously to canalize, to guide the learning mechanism. It will be completely useless to have a learning mechanism such as imprinting if every kind of possible information about the characteristic of your mother hand are provided in advance as an innate memory. What is useful instead is to guide the learning process in such a way to make it quick, fast learning. So consider the situation from the point of view of a newly hatched chick. You are out from the egg and there is a lot of things moving in the world. So natural selection acted in such a way to say, so to speak, to the young chick, please ignore, to focus on stimuli like completely rigid motion, say rolling stones. And on the other side, ignore objects like random motion, leaves moved by the wind, and focus instead in semi-rigid motion, which is a general characteristic, obviously, of all vertebrates. So it could be observed in a walking hen, your mother, but also in a walking cat. Doesn't matter, because obviously if it happens that there is a cat waiting for you when you are out from the egg, you are a dead chick. Well, that could be an interesting etymological story, and you can notice that, of course, if we look at the phylogenetic tree, the phylogenetic distance between humans, mammals in general, and birds dates back to about 300 million years ago. But there are evidences that this mechanism could be quite general and shared among vertebrates. And in fact, my colleagues in Padua, developmental psychologist Francesca Simeon, decided to test human newborns using exactly the same stimuli we use with chicks. So no human figures, but hen, chicken walking on purpose, of course. And what she found was that, yes, indeed, human newborns, a few hours old, have exactly the same preference for biological motion as newly hatched chicks have. And this makes us to consider again the story of the preference for the head and neck region in the young chicks. And we wonder whether a preference for face-like stimuli similar to that observed in human newborns could be observed in newly hatched chicks. You can say, well, chicks don't have a face. But if you filter high spatial frequency, you can see that there is the typical face-like appearance with the inverted triangle with 3 blobs high contrast, relatively higher with respect to a contour. So we look for spontaneous preference in completely naive chicks for stimuli like this one, face-like stimuli, looking at the first stimulus approach or looking at the time spent close to one or other of the 2 stimuli. And we found that naive, visually naive chicks have a striking preference for the face-like stimulus. And we performed several experiments comparing different conditions, including this vertical aligned set of elements or Picasso's-like, or stimuli with similar symmetry, but different organization with respect to the top and bottom position. This was done in particular to consider an alternative hypothesis which has been put forward, namely that the preference for the face-like stimulus could be related to the asymmetry in sensitivity in the retina of human newborns among the upper and lower meridian, and the idea that the preference for face is just the byproduct of a top-heavy preference. You can see this is not the case here because there is more elements in the top than on the bottom, but still they have a preference for the inverted triangle configuration, the face configuration. And we also studied face preferences using stimuli that were devised, I think, by Giorgio Vallortigara, in which naturalistic faces were compared with visual noise with similar spatial frequency, luminance, and color content. And we compared directly human newborns, they were tested by Mark Johnson in his lab, and chicks in our lab, and you can see that there is exactly the same preference also for naturalistic faces. So this set the stage for an investigation, a long-range investigation of animacy detectors, that is, subtle cues that serve to signal to young organisms the presence in the natural world of animated creatures, which is very important, the building block in a sense for the building of a social brain. So we investigated several of these. One is, of course, the idea that is associated to the name of Aristotle, that what is feature, the characteristic of living organism is self-propulsion, the ability to be self-moving. And to test this, we imprint chicks on an event in which an object, say A, move and then stop, and the other object B start moving, as in the classical launching effect of Albert Michot, and then looking for preferences for A or B object in naive chicks. And the idea is that one of these objects is self-propelled and the other is instead moving as the result of the collision. Obviously, color were counterbalanced, and so the direction of movement, and we performed a large series of control experiments. I'll show you some. For instance, this is the classical traditional launching effect. Could be that the chicks have a preference for the stimulus which is moving first. This can be checked, for instance, by changing the kinematics of the stimuli, namely moving first stimulus B and then stimulus A. Obviously, in this case, there is no collision, there is no physical contact, but this can be checked with this condition in which one object is moving and then it stops, but at this point there is a delay. And the delay is enough in order to have the impression, at least anthropomorphically for us humans, that there is no longer a causal mechanism producing the physical movement of the other, but even that the second object is moving as self-propelled. And the final control condition was one in which we tried to disentangle between the effect of causal agency and the effect of self-propelledness. So in this case, the stimulus is appearing from behind a screen, so because of this occlusion. So you cannot say whether it is really self-propelled or it is rather moved by another object, for instance, but the physical contact is still there. Well, to make short a long story, what we found was that only in the first condition there was a significant preference. So it is not the first object to move important, not the object initiating contact, not the object having a causal role. What is important is the fact that one object is self-propelled, is moving by itself, which is equivalent to say that it has in Aristotelian terms, an internal energy source, so to speak, as it happens with animated creature. And so it seems that chicks use these criteria as a way to select what is more likely to be a mother in a natural environment. And with Francesca, we try similar experiments in human newborns using preferential looking, and we use a sequence in which an object is visibly self-propelled, move, and then disappear occluded by a screen, and another condition where the object is appearing as the result of a disocclusion, so you cannot say whether it is self-propelled or not, and then stop at exactly the same position of the starting position in the other condition. And you see that there was a preference in human newborns for the self-propelled event. We investigated a variety of other animacy cues. For instance, self-propelledness is just one example of a more general mechanism which is associated with changes in speed, a condition in which the change in speed is extreme from an object which is standing still and then start to move. But even an object which is moving and then suddenly change speed is usually, at least to human adults, perceived as more animated than an object which is moving at a constant speed. So we wonder whether naive chicks are attracted by objects that change their speed. So we compared a condition with a constant speed like this with a condition in which a similar artificial object shown on a monitor screen is moving with a suddenly change of speed. And we found that, in fact, naive chicks have a striking preference for the first object. You can say that this is simply because they are attracted by novelty. But the point is that this novelty is related to the presence of objects that move at different speed, or what is crucial is perceiving the exact moment in which you see the change of speed. And to check for that, we compare, for instance, conditions in which there was a non-visible change of speed, like this one. So there is no novelty in this case, but because the velocity of the stimulus is changing, but you haven't seen the very moment in which the object has shown an acceleration, for instance, change in speed. And this seems to be, in fact, the crucial point, because still there is a preference for the one in which you can see, perceive the exact moment of the change of speed. Or similarly, if you have a series of occluders in such a way that the speed changes can be produced or in the visible part or the invisible part of the trajectory, or for instance with two constant speed, again, we can show that this was crucial for the chick's preferences. So for instance, two occluded speed changes and a constant speed were not producing any difference in this case. And another example of this sort of animacy cues, which seems to be inborn, immediately available to a completely naive chicks, are related to the constraints associated with the bilateral body plan. And there is a preference which has been originally described in human infants by Nikolai Hernik and Ergo Sibra, for objects that maintain their orientation with respect to the direction of elongation. And we tested this in young chicks with stimuli that were moving, maintaining their direction of movement with respect to the direction of the major axis, or stimulus that violated this sort of congruency, moving in part along the orientation of the major axis and in part along the orientation of the lower axis. Again, chicks show strong preferences for this kind of movement in which there is congruence in the organization of the bilateral organization of the body and the direction of the movement. Okay, this is a quite quick summary of of the overall behavioral evidence we collected with newly hatched chicks, but also with human newborns, related to the existence of this sense for animacy in very young creatures. In more recent years, we dedicated our research to try to understand the neural mechanisms underlying these abilities. The ideal condition would be obviously to study the neural mechanism directly in humans, but there are, as you can imagine, limitations, both ethical and practical. Nonetheless, recently we tried some experiment with human newborns we have a lab in the local hospital with an EEG and a FNIR setup, and we investigated preferences for face-like stimuli in human newborns using face-like stimulus, an inverted face-like, non-face-like actually stimulus, or this sort of Picasso's like face. And we use a frequency tag paradigm. That is, the stimuli were presented at a frequency of 0.8 Hertz with respect to visual noise, something like this sort of stimuli. And this is the face, and this is the non-face, and finally you can see the Picasso's face. I can show you an example of the stimulus presentation and recording. We can violate the privacy of these newborns because she is actually the daughter of one of my postdoc that kindly give us the permission to reproduce this movie. And these are the results. As you can see, the power spectrum of EEG responses was strikingly higher at the 0.8 frequency, at the tagging frequency for the face-like stimulus with respect to the non-face and the Picasso stimulus. And Marco Buiatti, who performed the experiment, also was able to reconstruct using some recent atlas of the skull and the brain of human newborns, the possible sources in the cortex of these EEG signals from the scalp. And you can see that very roughly, the same occipito-temporal area that are involved in face processing in human adults seems to be already active. These are 60 hours on average human newborns. But there is also quite a lot of activity here in this medial part that we suspect we cannot obviously be sure with the EEG, but we suspect is mostly associated with activation from subcortical areas and in particular from the colliculus. One interesting aspect is that obviously you can say, all right, these are very young newborns, but you cannot be sure that they have no experience with face. They certainly have probably some experience of the face of the mother, and therefore it could be that this is not an innate response, but it is rather some sort of very quick, very fast learning. It could be, but I am not very convinced, because if you look at the age of newborns in terms of hours, which show a certain distribution, obviously, and if you correlate the face-like pattern response with the aging hours, you can see that there is a striking negative correlation, which means that if there is an effect of experience of being exposed to natural faces, such as that of the mother and of the personnel and the people in the hospital, this is to reduce the original face-like preference. So I suspect that the preference for the face-like stimulus is actually similar, or maybe an example of what ethologists would call supernormal stimulus. You know, probably classical work by Nico Tinbergen showing that chicks of gulls that peck to the beak of the parents in order to obtain regurgitation of food in such way to obtain some feeding, when presented with different kind of dummies, one more zoomorphic, more similar to the actual appearance of the parent, and one composed simply by a pencil with red and white stripes. They would prefer the pencil because this is for some reason a sort of platonic general representation of the beak, which is more effective as a super stimulus. And I think that the face-like stimulus is a similar supernormal stimulus. It serves to attract the attention of the young baby to stimuli in the natural environment which are more likely to be a naturalistic face in such a way that the learning mechanisms can operate to learn the characteristics of the natural faces. Obviously, you know that we have, because of this animacy mechanisms, very sensitive to faces, and we can see faces everywhere. This is the phenomenon of pareidolia, and we see faces in the trees, in the peppers, in the Twin Towers. This is the famous demon devil face from the smoke of Twin Towers. And recently, Taubert et al. Showed that experimentally that monkeys have pareidolia because they usually prefer to look at the faces of conspecifics rather than to artificial objects. But if artificial objects are organized in such a way to produce the appearance of faces to be pareidolic, they look at this with the same, more or less, frequency as that of naturalistic images of conspecific. My favorite example is this one, and this is the very famous chick of the standard chicken. Okay, there are, as I said, limitations in the possibility in terms of the spatial and temporal resolution which is allowed looking at the human brain. And this is the reason why we usually are interested in using animal model systems such as chicks. This is the brain of a chick. It is quite large, in fact. A newly hatched chick has a brain which is more or less the size of an adult mouse. And the general organization is similar to that of all vertebrates. This is a telencephalon for brain. This is the tectum, and this is the cerebellum. Obviously, we were not completely naive with respect to the brain areas that could be involved in animacy predispositions. In particular, we were interested in the already described social behavior network. There is a network of brain areas which are largely shared among vertebrates and so very ancient, which are involved in the adult animals, and mice are being studied in particular, in a variety of social behavior. And this area include the amygdala, several nuclei of the amygdala, the lateral septum, the medial preoptic area, and other regions such as the anterior and the ventromedial hypothalamus. And they are differently involved in the adult, for instance, in sexual behavior, in territorial behavior, particularly the lateral septum, and the part of the amygdala in sexual behavior. Gregarious and colonial behavior involve both septum and area of the hypothalamus, and so on. As I said, these regions show similarity in the pattern, in the connectomic organization, both in mammals and in birds, and so they are certainly derived from some ancient vertebrate chordate ancestor of both mammals and birds. I can give you an idea of the position of these brain regions. We are trying to prepare an atlas of the chicken for brain for other purposes, for fMRI studies, and using our MRI facility, we obtain some image and the position of the social behavior network nuclei are shown here, and we will concentrate in particular on these three areas that we found to be important, as I show you in a minute, namely the middle preoptic area, which is shown in red, and the lateral septum, which is a very ancient structure of all vertebrate brain, and the nucleus taeniae, which is homologous of the medial part of the amygdala in the mammalian brain. If you are more familiar with the traditional coronal section, just to give you an idea, this is the position of the lateral septum, which is here, and there are several subdivisions which are of interest for neuroanatomical studies. And these are the arcopallium, which is more lateral, and this is the position of the AMM that I mentioned before. This is not subcortical, it's a cortical, equivalent of a cortical structure, and it is involved in the memory for imprinting. And finally, as a control, we also look at the medial striatum that we know, more rostral, involved in learning, but not apparently in imprinting. So the technique we use is a very well-established technique in neurobiology, namely the expression of so-called immediate early genes, that they can be considered as activity marker of neural activity by immunohistochemical staining of the product of a gene, the CFOS, that the protein, the CFOS protein, is expressed in neurons that have been associated with neural activity. So we can see which neurons are stained, and using automated procedure, we can count these neurons. So the level of spatial resolution is the level of single cells. And basically the procedure is something like that. We have two groups of chicks, two or more, and chicks are presented for a very short period of time with a stimulus that could have an animacy cue, say face-like appearance, or not, with similar physical characteristics, so an inverted face-like stimulus. And the stimulus is presented for a time which is too short in order to produce imprinting, usually 10 minutes. And then the animal is sacrificed, and we look for expression of CFOS in different areas of the brain to see whether there is selective activation associated with stimulation produced by animacy cues with respect to non-animated cues. So we started with something very general and very rough in a sense, providing chicks with a complete real conspecific containing all sorts of possible animacy cues. And so there is an experimental chick looking to another chick and another baseline chick looking at an empty environment. So olfactory information, acoustic information is shared, but visual information, which is very rich in this case, is limited to just one chick. And what we found was selective activation, expression of the immunoreactive neurons in the septum and in the arcopallium and nucleus taeniae, not in the medial striatum, not in the AMM as expected. This is a very obviously complex stimulus, so we try to make something more simple, trying to disentangle the purely dynamic cues from the static ones. So we presented this time either an alive chick or a rotating dummy chick. And this is the experimental condition. You can see they are both moving, and obviously the stationary cues are exactly the same. There is a hand and neck region, but the kind of motion is completely different. So biological motion, change of speed, self-propelled is provided by one of the two stimuli, but not by the other. And what we found in this case is that there was selective activation only in two areas, and only in the right hemisphere for the dorsal septum, and only in the left hemisphere for the preoptic area. No in ventral septum, no arcopallium, no nucleus taeniae. Conversely, we try to disentangle which brain area could be, this is still a complete or relatively complex stimulus, so we try to make something more simple. Extremely simple, so we presented chicks this time with a red disc moving with speed changes, all moving at a constant speed. So a very, very subtle difference. And what we obtained this case, again, was a selectivity of response in the lateral septum confined to the right hemisphere, and no other statistically significant difference. This is just an asymmetry, but not specific of the stimulus in the AMN. In order to see which brain region is important for stationary stimuli, we use the classical stimuli of Gabriel Orr, namely a canonical stuffed hand and an N which was completely scrambled. And what we found in this case was a selective activation without brain asymmetry, only in the nucleus taeniae, so in the equivalent of the lateral amygdala. So to summarize, when all cues for animacy were combined, the septum and arcopallium region seems to be involved. When information was limited to stationary configuration associated with animacy, the nucleus taeniae seems to be important. And when only motion cues were available, and in particular for speed changes, the septum and the preoptic area were important. More recently, we have been trying to investigate biological motion as conveyed by point light display. And here the story is a little bit more complicated because, first of all, we found that if we compare the walking hand with the upside down version of a walking hand, you can observe that there is a classical inversion effect. It's very difficult to recognize the animal, the shape in this case, but it is not the equivalent of the face inversion effect, because in this case, what is crucial is not the fact that the orientation is not the canonical one, but rather the fact that there is psychophysical evidence for this, the fact that the direction of gravity is violated, typical movement with respect to the direction of gravity. So in chicks, we found that if we look at the way in which a chick is oriented with respect to the apparent direction of the point light display, for instance, if during the first 4 minutes, we look at the overall consistency of chick's body orientation with the walking direction, say in this case from left to right, and then after minute 4, from right to left, you can see that the chick change orientation and maintain they align with respect to the direction of the apparent movement of the hand. But if the point light display is inverted, then they are completely random, which is interesting because the stimulus is still attractive. It is biological motion, of course, but they are no longer able to use the kinematics of the stimulus in order to recover, to obtain the direction of motion. And what is even more interesting is that probably this is associated with brain asymmetry, because if you look at the particular direction of movement and therefore to the eye that the chick is using, the effect is apparent only for rightward motion, which means that they were using the left eye and therefore the right hemisphere in order to exhibit this preference. Please remember that in chicks that have laterally placed eyes, there is complete decussation of the optic chiasma, and there is no corpus callosum in birds, which is a characteristic of the mammalian brain. So information entering each eye is largely processed by the contralateral hemisphere. So very recently, this week, we are trying to investigate the neural mechanism by C-FOS expression. So chicks are presented briefly with random motion or with walking hand. And at the start, they are, before exposure, moving in the complete darkness in a running wheel. And then they are tested. They are presented with the stimulus for a few minutes and then sacrificed. And the results are shown here. There is still the septum, which is activated as expected, non-nucleus taeniae, no preoptic area. That is interesting because it seems to be different from speed changes. But there is also activation in the AMM. This is the first case in which there is this sort of activation. And we have to really ponder about this and what it means. It could be that this very rich stimuli started the imprinting procedure, which is in the IMM initially very early, starting from already a few minutes of exposure. That would be a possibility. We also look at as obvious in a sense, if there is an effect of oxytocin. You know, this is a very hot topic for research in this year. So this is being called sometimes the love hormone. And there is a homologous of the oxytocin in the bird brain, which is called mesotocin. So we injected chicks intracranially with mesotocin, and you can see here that with respect to the saline, there is indeed an increase in the spontaneous innate preference for face-like stimuli. Next step would be trying to do lesion experiment. We expect that lesion in the septum, for instance, preferences for speed changes and biological motion would disappear. And the other step is to try to do single cell recording. We have some data for that. We recorded certain number of cells, not a large number yet, in several brain regions. It seems that indeed there are single neurons in completely anesthetized chicks that show activation, selective activation for face-like with respect to non-face-like stimuli. I describe mainly only actually animacy detectors related to social life. But as you can imagine, animacy detectors are relevant also to reveal the presence of different kind of creatures in the environment, including also predators, for instance. So recently, we look at the effect of a simulated predator in the form of a looming stimulus, that is a disk that suddenly would enlarge in size, or a sweeping stimulus, a disk which is simply moving with a translatory motion over the head of the chick. These are completely naive chicks that without any kind of experience immediately show different and adaptive responses, which is very quick escape responses in the case of looming, because the predator has spotted you, or freezing and immobilization in the case of the sweeping. And this is shown in this video, in which you can look at the different kind of reaction. This is the response to the looming stimulus. You see, it is presented on the side, but it is actually over the top of the animal. And then you will see the quite different response to a different kind of treat, namely the sweeping stimulus, that when noticed, make the chick stop, freeze, and couching down, which is quite typical. Okay, I think I can skip the details of these experiments. And I just want to note as an historical curiosity that you know probably that the freezing response, the extreme freezing response to pretend to be dead is a anti-predatory response which can be observed even when you are in the clutches of a predator. And it is the basis of so-called animal hypnosis. This has been described, to my knowledge, for the first time by Father Athanasius Kircher, a Jesuit priest, in 1648 in the book Ars Magna Lucis et Umbre. There is a chapter titled Experimentum Mirabile. In Latin, this means wonderful experiment, in which he drew a line on the floor, and it was able to obtain hypnosis of a chicken. Actually, the line is irrelevant. What is important is simply to block the animal with the hands for a few seconds. This simulate the attack of a predator and is producing the tonic immobility in the chick. Okay. What can we do with this super chick, with this particular model system? The general idea is that these animacy detectors are, in a sense, the building blocks for social brain. They are the initial responses that, guided by genetic mechanisms, that serve to guide, to canalize the learning mechanism, learning about the characteristic of the social environment. So if something went wrong at the start and these animacy mechanisms are not operative, then you can expect disorders in the neurodevelopmental mechanism. Disorders, for instance, like autism spectrum disorders. Maybe you are familiar with this drug, the valproic acid, which has been used for a long time as a mood stabilizer and also as for the treatment of epileptic patients, until it was discovered that it is quite dangerous for mothers because there is a high risk to have a progeny with autistic spectrum disorder. And so valproic acid is now very much used for research, and we wonder whether administration in the egg, we administer valproic acid on day 14 in embryo, could have an effect on animacy predisposition. And we tested preferences of chicks injected with valproate for both the preference for the head and neck region, comparing canonical and scramble hand, and also for changes in speed. So very different, very naturalistic and very unnatural condition. And we found that indeed, chicks treated in the egg with valproic acid completely abolish the preference. The dosage was very low, so there was no important effects on behavior. And in fact, for instance, imprinting was totally unaffected. These chicks were able to imprint, for instance, on the scramble or on the canonical, but they have no any preference for one or other. So the learning mechanism is intact, but the innate preference that guide the learning mechanism is abolished. As you know, valproic acid is important also with respect to the issue of sensitive periods. That is that during limited time during development, the effect of experience on brain function and behavior is particularly strong. But this is occurring obviously for imprinting in chicks. There is a period, usually first 3 days, after which imprinting is no longer possible. But recently it has been found that administration of a thyroid hormone, T3, seems to be implicated in the timing of the sensitive period. In chicks, imprinting causes a rapid inflow of T3 in the brain, particularly in the dorsal pallial region, that is the AMM and other associated region, through converting one enzyme, which is called DIO2, which is localized in vascular endothelial cells. If chicks are given endogenous T3, endogenous T3 level in the brain peaks at around peri-hatching period, and this is shown here. You can see that there is this peak. It is a little bit before hatching, probably favoring acoustic imprinting. We don't know about that. However, if in non-imprinted chicks whose sensitive period is ended, we administer exogenous T3, this allows imprinting to occur. So there is a reopening of the sensitive period for memory formation. Obviously, reopening of sensitive period by pharmacological agents has been discovered for a variety of other phenomena other than imprinting, say ocular dominance, and also absolute pitch in humans by Judith Gervain and Takeo Hensch, for instance. But the interesting issue for us is, could it be that there is also a sensitive period for social predisposition, and then we can affect this sensitive period by thyroid hormone T3? So we investigated again speed changes as a paradigmatic example of animacy cues. And then first we look whether there is a sensitivity window. So we tested chicks on day 1, and this was a replication of a previous work, and they show the preference for the speed changes. And then on day 3, and on day 3, the preference is completely abolished. Then we try to see whether blocking T3 is blocking the preference on day 1. So we administer a particular drug, which is IOP, which is an acid that blocks the expression of T3 hormone. And as you can see, chicks injected with IOP didn't show any preference, whereas those with the vehicle showed the usual, the ordinary preference. Then we try the next step, namely to make a resurgence of the preference on day 3. And there is a complication here because on day 3, sex differences become apparent in the behavior of chicks. So you can see that, yes, administration of T3 in female make reappearance the preference, but quite paradoxically, the vehicle showed the reverse effect, and males were some way midway and not very clear results. This is surprising because obviously in the vehicle, we expect no physiological action of the saline solution. So the only possibility is actually that the pain associated with the injection, the needle, is making in some way this effect, is producing some sort of avoidance of the more animated stimulus in female. And in fact, if you look at the responses in the absence of any substance, so in absence of any injection, and so any needle, any pain, you can see that there is not any expression of a preference in, on day 3, but in females injected with T3, there is the expression of the preference. The other line of research in which we are trying to test this idea of animacy detectors as building blocks of social brain is to look directly at the behavior of human infants and children with autistic spectrum disorder, and to use our finding as a way to make early diagnosis of autism. As you know, obviously the diagnosis is possible only starting from year 2, 3, which is very late, but we can circumvent in research this difficulty by looking at a particular condition that is studying so-called high-risk autism children who are siblings of individuals, usually males, that have been already diagnosed with autism. And there is a network in our country, and as well as in other countries, of course, and they alert us when there are available, with the permission and the interest of the parents, these high-risk newborns, and we tested them when they are between day 6 and day 10. So obviously we cannot test them in the hospital, it would be too much complicated in terms of ethical permission, so we wait to test them at home of the parents. And we have a mobile lab limited to the behavior up to now, but we hope to be able to do also EEG in the near future. And basically we test them with exactly the same test we perform with our young chicks. Using looking time preferences, we test these newborns for their preferences for face-like or non-face-like stimulus, for biological motion and random motion or rigid motion. You can see here the example for the face. And what we found is that whereas low-risk individuals show the expected preferences for face-like stimulus, no preferences observed in the high-risk individual. And low risk show the expected preference for biological motion with respect to random dot motion, but not so the high-risk individuals. And also for rigid motion. Actually, for rigid motion, there is a slight tendency, not significant actually, for high-risk individual to show a preference for the rigid rather than to the biological motion. Well, these results might allow different interpretation. One interpretation is that there is, for genetic reason, probably a lack, an absence of the mechanisms for detecting animacy cues in the natural environment. The other possibility is that these mechanisms are simply delayed in their development. And some follow-up, we have the first data for the follow-up at 4 months suggests that indeed the problem could be a problem of development. Look at these results. For instance, for the face-like stimuli, as expected in the low-risk individual, there is a preference in newborns, but the preference disappears at 4 months. But the reverse seems to be true for high risk. This is not significant for face, but it is significant for averted versus gaze. They show a preference, and also for biological motion. They show the preference for biological motion versus random motion at 4 months when it was not there when they were tested as newborns. So it could be that these individuals are in a sense out of phase, even with respect to the mutual dialogue of gaze, which is observed soon after birth between the mother and the baby, and that when they start to look actively to the face of the mother, to the face-like stimulus, to the biological motion, and so on, it is in a sense too late, and also the mothers would realize that there is something wrong, something which is unusual in these babies. Okay, so I think I can stop at this point, I just want to add thanks to all my postdocs that have worked on this very long project, which has been funded by the ERC. And so thank you very much also to ERC. And in general, this time is important to stress, thank you very much to Europe. And thank you very much to all of you for your attention. And of course, I am now ready for answering your questions. Thank you.
Everybody, let's thank Professor Vallortigara for his talk by clicking on a button that shows clapping hands or by doing this. And now it's time to ask questions. And the way that's going to work is if you move your cursor down, you see a participants button. If you click that, the list of participants comes up, and there you can raise your hand. And if you do that, I know you have a question, and then I'm going to, if there's multiple people raising their hands, I'm going to pick one of them and let that person unmute themselves and ask a question, and then our speaker will respond. And you can also see in the chat somebody's pointing out that where the raise hand button is depends on your version of Zoom. It might be in the reactions menu along with the applause. It is in mine as well, actually, so good point. So please raise your hand if you have a question. Okay, I see some hands raised. I apologize in advance if I mispronounce your name. It's my fault. So I would like Kylie Hamlin to ask their question. So I'm going to ask them to unmute.
Hi, great. I don't think I can do my video, but that's fine. Thanks, Giorgio. That was really wonderful. I'm just curious about your sense of the sort of, it seems that your argument is that these animacy detectors are key sort of core aspects of early social detection. And I'm wondering to what sense you think that there may be additional mechanisms that are somewhat higher level than animacy detectors that are also sort of foundational, or whether really everything follows from these basic mechanisms. Thank you.
Okay, can you hear me? Okay, thank you very much. Thanks the organizer for the invitation. Thank you very much, and thank you, Dr. Hamlin, for the question. I think that the evidence we have got with EEG recording in human newborns would suggest probably that the mechanism we are discussing, at least for humans, I don't know for sure in other species, could be also in preparation of more cortical depending aspects related to not just animacy, but also maybe agency. So in this sense, I suspect, but I have no any direct evidence for this, apart from the quite surprising fact that there seems to be already cortical activation in response to phase-like stimuli, not just and specificity, and not just only at the subcortical level, at least in humans. I know Obviously, source reconstruction from EEG is in some way controversial, but I think that the evidence is at least intriguing in this regard.
So next question is by Jean-Remy Hochmann, who I will now ask to unmute.
All right, good evening. Thank you very much for the talk. I have actually two very quick questions about the autistic, well, the high risk for autism study, which I'm sure is it's a beautiful study and probably very hard study to run. But I have the question about the controls of the studies. So the first very quick question is, do you have any way to have another population as control? And I was thinking, for instance, of infants who would be at risk of attentional deficits. And I was also thinking that, so if I understood correctly, the high risk infants are infants who have a sibling that is autistic. So it could be that the delay in the emergence of preference for social stimuli suggests a role for experience, and maybe those children have less attention from the parents because of an autistic sibling. So I was wondering if you considered such an explanation.
Yeah, sorry, I forgot to activate also the video. Hello, good evening, Jean-Remy. Thank you for the question. Yes, with respect to the control, no, we have only newborns tested separately as normal development. So we don't have another condition in which there is some sort of other different pathological conditions. So we have simply normal development newborns that were used as a control group. With respect to the effect of experience and the role of experience in having been raised by parents with sort of experience related to autistic spectrum disorder, well, I would say that I would expect more attention in a sense and more dedication and more experience, exactly because these parents were in some way alerted and very worried about the condition of their children. But again, in the absence of direct testing, it's difficult to say.
Our next question should be from Gil Diesendruck.
Hi, yes, hello all. Thank you very much for the talk. I was a bit puzzled, surprised by the argument you presented as to why the animacy detector doesn't discriminate between conspecifics and those that do not. In the chicks experiments in the beginning, you later talk about the possible distinctions between predators and non-predators. And so I was wondering if you have any hypothesis about when these kinds of sort of, functional sensitivities as to whether I should approach something that I detected as an animate versus whether I should avoid, run away from something that I detected as an animate. When does that come online? And why, if at all? I mean, shouldn't that also be built in to the system?
Well, I think that in the case of social responses, those mediating affiliative and the following responses in chicks, the crucial point is that the role of this predisposition is basically to reduce the time for learning. In other terms, as I said, it would be completely useless to have a learning mechanism if you provide an organism in advance with all the information. So if you say to the chick, look, this is a hen, and the hen is moving in this way, and the hen has this appearance and so on. In this case, if all is provided as a genetic memory, learning would be completely useless. So the function of this predisposition with respect to learning, obviously, is simply to make restricted the range of stimuli to which you are more likely to direct, for instance, following responses. For responses which are more fast and quick, such as anti-predatory responses, the evidence suggests that in this case, there are very, very simplified forms of stimuli which are highly effective, such as looming. For looming, we know that there are neurons from a long time in the tectum that respond directly to looming stimuli. And also human newborns are sensitive to this aspect. Consider that these are very quick and very fast response that may be apparent at the start for any kind of motion stimuli. But then, if after immobilization, freezing responses, there is no reason for further danger, the animal can look at the stimulus and start to decide whether to approach the stimulus on the basis of whether this particular kind of motion is associated with a predisposition for approaching and showing the following response. So I think that speed and sequence of proper behavior from anti-predatory to following responses could be important. The same is true also for face-like stimuli, because, for instance, there's a long-standing and unsolved issue concerning what is important really with respect to this face-like stimuli. If, for instance, the eyes alone would be important, because there is quite a lot of evidence showing that the configuration of horizontal arranged dots corresponding to a potential frontal eye is aversive, is inducing an anti-predatory response in a variety of animals. So one possibility is that some of these animacy cues were originally derived from mechanisms more general to respond to animated creatures, like anything of living creature, and in particular predators, and then readapted for social function. That could be the case, for instance, for the origins of the responses to face-like stimuli. One very interesting paper which has been published recently on PNAS by a former postdoc of mine, Elisabetta Versace, has shown, for instance, that face-like preferences are observed in tortoises. Tortoises completely lack any kind of parental care. So clearly the preference for face-like stimuli is not strictly associated with social responses, seems to be something which has a more remote origin to detect animated things, living things. And then they can be used in a social context or in an anti-predatory context, depending on the ecological condition of a particular species. Or at least that's my hypothesis.
And we have a question from Yuri Gergely, who I will unmute. Yes.
You hear me now? Thank you. Well, thank you for the really wonderful talk. My question relates to my memory of the infant research on the face pattern preference, like Teresa Faroni and Gergely Cibra, Mark Johnson, where the white background, like this white sclera, red pupil, which is a human adaptation, it seems, has been an important determinant of the early preferential response. And I wonder whether, this is a kind of an add-on of human evolution to this more basic response to like an animate predator significance possibly, or whether you have tested, or whether this kind of difference between the the background and the foreground of the white and the dark is also there in chicks. I don't hear this.
Okay, can you hear me?
Now I can hear you, yes.
Okay, thank you very much for the question, and hello. Yes, we did test this issue. I have no time. It was already a very long talk, so I omitted this part. We tested chicks for face-like stimuli with invert contrast. And what we found was that there was, depending on the experimental condition, or complete lack, abolishment of any preference, or active avoidance when the contrast was reversed. And we suspect that avoidance was exactly related to the fact that in this case, there was a response to a predator-like stimulus rather than to a social stimulus. So yes, there is quite close similarity with the data of Teresa and Gergo with human newborns.
So don't be shy. Do we have any further questions? I mean, if you are shy, that's also fine, because the concept of the conference is you can ask your questions later, ask them in writing. If there are no questions, I would like to again thank Professor Vallortigara for his time and for coming here today with a little hand symbol. And
thank you.